Hello there, and welcome to the Sleepy Scholar. Tonight's journey takes us out into the vastness of the unseen, the utterly essential, and the surprisingly beautiful world of the microscopic. We are preparing to quiet the clamor of the large and listen instead to the deep, silent symphony of the very, very small, where cells hold ancient memories and water molecules dance in infinite, complex choreographies, a place where the familiar rules of our everyday reality gently begin to dissolve. Allow the magnitude of this unseen universe to wash over any worries you carry, for here, beneath the threshold of ordinary sight, existence is an enduring ballet of precision and patience, a constant, tireless unfolding that requires nothing from you but your quiet attention and your willingness to rest. If you enjoy these quiet explorations, consider subscribing so you can easily return here whenever you need to rest. For now, get comfortable. Let your eyes grow heavy. Let's begin. The diameter of the smallest known bacterium, Mycoplasma genitalium, is around 200 nanometers, requiring incredible patience to observe. Before the advent of modern electron microscopy, the true scale of life was largely confined to philosophical speculation, with scientists only able to infer the presence of agents smaller than light allowed them to see, standing like quiet observers at the very edge of an unimaginable ocean. This tiny organism, which carries one of the smallest genomes of any free living life form, acts as a profound testament to the efficiency of evolution, demonstrating that life can sustain its vast complexity and purpose with the barest minimum of genetic instruction, essentially performing a kind of minimalist molecular ballet. The genome itself, a ribbon of nucleic acid, unfolds its information with a kind of stately, unhurried grace, guiding the cell's processes of replication and survival against the chaotic backdrop of its environment, a perfect, self-contained universe of function. To gaze upon its size is to experience a gentle reset of perspective, realizing that the most powerful engines of existence often operate far below the din of our sensory world, holding their vast and quiet power in the realm of the truly minute. The human body contains trillions of microbial cells, collectively known as the microbiome, which softly governs much of our physiological peace. Long ago, these countless microscopic companions began their tenancy within us, not as invaders, but as silent, necessary partners, evolving alongside our own lineage in a relationship of deep, unspoken mutual reliance, a quiet, ancient treaty written in the language of biochemistry. These vast communities, dwelling primarily within the intricate winding landscapes of our gut, help to dismantle complex foodstuffs that our own enzymes cannot manage alone, transforming them into vital nutrients and gentle energy that flows through us like a calm, sustaining river. Furthermore, this microscopic peacekeeping force acts as a protective barrier, teaching our immune system which foreign entities are welcome guests and which are truly disruptive visitors, instilling a profound balance that prevents unnecessary alarm and chaos within our inner world. Much like a meticulous unseen gardener tending to a sprawling sacred estate, a single drop of ocean water can contain millions of phytoplankton, which are the silent, breathing engine of our planet. These minuscule, plant-like organisms, often overlooked in the consideration of global majesty, drift passively in the sunlit upper layers of the vast, cradling oceans, absorbing sunlight with a gentle, enduring dedication that converts simple water and carbon dioxide into the very foundational elements of life. Through the process of photosynthesis, performed on an astronomical scale that dwarfs human ambition, these quiet beings release immense quantities of oxygen into the atmosphere, allowing all terrestrial life to partake in the deep, sustaining breath of existence, connecting the smallest organism to the largest. When they eventually complete their gentle cycle, their microscopic bodies sink slowly into the profound darkness of the deep sea, carrying carbon with them, sequestering it for long geologic ages, acting as the Earth's most effective and unhurried regulator of the planetary climate, a timeless, patient, and essential force of transformation. The movement of a bacterium, often achieved via flagella, is a sophisticated, whirling mechanism of quiet molecular propulsion. Imagine a tiny, elegant motor, not driven by gears and fire, but by the quiet flow of protons across a membrane, spinning an external tail with a speed that is utterly confounding for something so small, allowing the cell to navigate its watery landscape with purpose. This flagellum does not simply swim. It performs a deliberate run and tumble routine, moving smoothly toward a pleasing chemical signal before momentarily stopping its rotation to randomly reorient itself, essentially executing a series of thoughtful, self-correcting movements across its microscopic domain, guided only by the gentle whispers of concentration gradients. This exquisite piece of biological engineering is an ancient design, perfected over eons of silent evolution, and serves as a humbling reminder of the profound complexity that dwells within even the most rudimentary forms of life, a quiet dedication to motion and survival that transcends our scales of comprehension. Mitochondria, the powerhouses of the eukaryotic cell, possess their own distinct circular DNA, a gentle echo of their ancient, separate origins. It is theorized that these vital organelles were once independent, free-living bacteria that were gracefully and quietly absorbed by larger cells billions of years ago, in an act of profound, mutually beneficial cooperation known as endosymbiosis, an ancient cellular embrace that fundamentally reshaped the trajectory of all complex life. This residual DNA, passed down almost exclusively through the maternal lineage, carries the quiet, indelible memory of their past freedom, silently directing the synthesis of specific proteins essential for cellular respiration, the slow and steady process of unlocking life's sustaining energy from simple sugars. The presence of these internal, semi-autonomous entities suggests that life's grand architecture is often built upon layers of successful partnerships, where separation gives way to integration, leading to a profound, restful efficiency that powers the thoughtful activities of every complex organism, including the slow, deep process of human contemplation. The nucleus of a eukaryotic cell is a membrane-bound sanctuary that cradles the precious, undulating ribbon of the organism's genetic destiny. Within this quiet, central chamber, the chromatin, the mixture of DNA and protein, is carefully organized into territories, ensuring that the vast, complex instruction manual of life can be accessed and utilized with a quiet, meticulous order, avoiding the chaos that would surely ensue from disorganization. The nuclear envelope, a double layer of lipids, acts as a soft, permeable boundary, studded with complex protein portals that govern the deliberate, selective exchange of molecular messengers, ensuring that the nucleus remains a zone of concentrated, deliberate action, shielded from the boisterous activity of the surrounding cytoplasm. This protective containment ensures the genome's stability and the measured pace of gene expression, allowing the cell to build and maintain its form with a serene, continuous flow of information, the gentle, guiding whisper of heredity playing out in perfect peace. An enzyme is a protein that serves as a profoundly efficient biological catalyst, gently accelerating chemical reactions without being consumed itself in the process. These molecular laborers operate with a quiet, stunning specificity, often possessing a uniquely shaped active site that perfectly accommodates only one type of substrate molecule, ensuring that the cell's intricate metabolic pathways remain distinct and orderly, like a massive orchestra where every musician plays only their appointed, harmonious part. Their action allows reactions that would otherwise take hours or days to occur in the blink of an eye, facilitating the rapid, smooth flow of energy and matter required for life's continuity, essentially eliminating the stress of slowness and allowing the organism to operate with a restful pace. The enzyme's ability to bind, transform, and release its substrate, then immediately prepare for the next task, demonstrates a tireless, graceful efficiency, a quiet mastery of speed and precision that underpins the entire, enduring structure of biological reality. The cellular cytoskeleton is an intricate, dynamic scaffolding network that quietly dictates the shape, movement, and internal organization of the cell. Composed primarily of three types of filamentous proteins, microtubules, intermediate filaments, and actin filaments. This internal support structure is not static, but is constantly being assembled, disassembled, and reorganized with a fluid, silent grace, allowing the cell to softly adapt its posture and respond to its surrounding environment. Microtubules, like tiny, quiet structural beams, guide the internal transport of vesicles and form the core of cilia and flagella, facilitating organized movement. Intermediate filaments provide enduring, stress-resistant tension, acting as the cell's quiet, tensile strength, and actin filaments enable movement at the cell's periphery, allowing for graceful creeping or profound shape changes. This entire, invisible framework ensures that the cellular world, though microscopic, possesses a powerful, organized architecture, lending an enduring, quiet solidity to the very fabric of life. The concept of Brownian motion describes the quiet, perpetual, random movement of microscopic particles suspended in a fluid or gas, caused by the continuous, unseen bombardment of surrounding molecules. This phenomenon, which can be observed under a high-powered microscope as a kind of gentle, ceaseless molecular jitter, provides profound, direct evidence of the existence and kinetic energy of atoms and molecules, revealing the quiet, constant thermal motion that underlies all observable stability. The random path traced by a colloidal particle is a soft, visual manifestation of the universe's ceaseless energy, a chaotic dance that, on the macroscopic scale, resolves into predictable laws, demonstrating how the immense clamor of individual movements gives rise to a profound, restful stillness when viewed in its totality. To watch this silent, tiny wavering is to observe the very breath of the universe, realizing that even in the absence of obvious external force, motion is an eternal, inherent property of matter, a gentle, enduring vibration that never truly ceases. Diatoms are single-celled algae encased in beautifully sculpted cell walls made of silica, often resembling tiny, exquisite works of architectural glass. These microscopic beings, which exist in incredible abundance in both freshwater and marine environments, construct their intricate, patterned frustules with a quiet, geometric precision that adheres to the strictest principles of natural design, achieving strength and lightness simultaneously when viewed under magnification, The detailed symmetry and fractal-like complexity of these shells evoke a profound sense of awe, suggesting that the drive for beauty and pattern is deeply embedded even in the most fundamental layers of existence, a silent artistic impulse that shapes the mineral world into living forms. Upon their gentle passing, their glass shells sink to the ocean floor, forming vast, deep sedimentary layers known as diatomaceous earth, which acts as a quiet geologic record of oceanic history, a long, enduring testament to the vast, quiet power of the smallest of lives. The process of osmosis describes the passive, unhurried movement of water molecules across a selectively permeable membrane, seeking a state of gentle, equalized concentration. This fundamental principle of biophysics is driven by the desire for equilibrium, a profound, unceasing molecular yearning for balance, where water flows from a region of higher concentration to a region of lower concentration, silently regulating the volume and internal pressure of every living cell. Without this quiet, continuous process, cells would either swell dangerously or shrink and wither, losing the internal turgor necessary for life's architecture to stand firm, demonstrating that the most essential functions of life depend upon the constant, gentle push toward a state of systemic peace. Osmosis is a beautiful, slow demonstration of nature's relentless pursuit of rest, illustrating that complexity arises not from force, but from the simple, quiet elegance of molecular mathematics. Prions are infectious agents, composed purely of misfolded protein, carrying no nucleic acid, which can quietly induce normal, healthy proteins to also adopt the aberrant configuration. This concept challenges the traditional notion of life, requiring genetic material for replication, suggesting instead a kind of molecular contagion of pure shape, where the profound and patient structure of a protein is the infectious element. The process is slow, insidious, and entirely silent, propagating an incorrect fold with a domino-like, patient inevitability that gradually disrupts the quiet, ordered function of brain tissue, leading to neurodegenerative conditions that unfold across a vast, slow arc of time. The very existence of prions serves as a profound, unsettling meditation on the power of molecular form, illustrating how a simple change in quiet, spatial configuration can become a powerful, self-propagating force, transforming the very architecture of consciousness with an unnerving, patient persistence. The cell membrane, or plasma membrane, is a double layer of lipid molecules, often described as a fluid mosaic, which acts as the gentle, defining boundary of the cell. This exquisite structure is not a rigid wall, but a dynamic, undulating sea of phospholipids, interspersed with various proteins, allowing the membrane to flex, heal, and adapt to the endless microscopic currents of its environment, while maintaining its profound integrity. The fluid nature of this boundary ensures that components can drift laterally, allowing for the gentle restructuring necessary for processes like cell signaling and the soft fusion of vesicles, embodying flexibility and strength in perfect, silent concert. It is the gatekeeper of the inner world, meticulously controlling which molecules are permitted to cross the threshold, maintaining the delicate, necessary distinction between the ordered, internal peace of the cytoplasm and the turbulent, external universe, allowing the cell to remain a sovereign, stable entity. The process of phagocytosis allows certain cells, such as immune cells, to softly engulf and consume large solid particles, acting as a quiet, protective internal cleansing system. This graceful act of cellular ingestion begins with the cell extending long, flowing pseudopods that slowly and deliberately surround the target particle, be it a foreign bacterium, a piece of cellular debris, or a dying cell, drawing it into the internal world with a profound, unhurried purpose. The engulfed particle is then contained within a membrane-bound vesicle called a phagosome, which fuses with a lysosome, introducing a collection of digestive enzymes that gently and thoroughly break down the captured material, neutralizing any threat and recycling usable components. This constant, silent consumption of the old and the dangerous is essential for maintaining the clean, restful organization of tissues, acting as an unceasing, dedicated patrol that preserves the internal peace of the entire organism. Ribosomes are the cellular machinery responsible for translating the genetic code carried by messenger RNA into the long, beautiful chains of amino acids that constitute proteins. These complex molecular factories, found both floating freely in the cytoplasm and attached to the rough endoplasmic reticulum, operate with a quiet, tireless dedication, reading the code in precise triplets and sequentially adding the correct amino acid to the growing polypeptide chain, a process that unfolds with rhythmic, stately precision. Each protein is constructed according to an exact, ancient blueprint, and the ribosome's action transforms the abstract language of genetic letters into the three-dimensional, functional architecture of life, a profound act of translation that is fundamental to all existence. The sheer volume and relentless, synchronous activity of these minute organelles within every cell represents an overwhelming, quiet industry, the constant, silent hum of creation, occurring in billions of tiny workshops within your own resting body. The concept of a biofilm describes a community of microorganisms, often bacteria, that adhere to a surface and encase themselves within a self-produced matrix of extracellular polymeric substances, which acts as a quiet collective shield. This gelatinous, structured home provides a profound, restful protection from external stresses, such as desiccation or the flow of antimicrobial agents, allowing the community to thrive in environments that would otherwise be too harsh for single, isolated cells, embodying strength through soft, unified architecture. Within this soft, communal structure, the organisms can communicate with each other using chemical signals, a silent language known as quorum sensing, which allows them to coordinate their behavior, such as activating gene expression across the entire population, acting with a unified, gentle purpose. The formation of a biofilm is a beautiful, enduring example of microscopic cooperation, illustrating how simple life forms find profound security and peace in creating their own carefully managed, self-sustaining environment. The structure of liquid water, at the microscopic level, is one of constant, transient hydrogen bonding, a ceaseless, flowing network of gentle molecular attachment and release. Although the water appears still and uniform to the naked eye, on the scale of molecules, the water units are perpetually forming and breaking weak hydrogen bonds with their neighbors, creating an ever-shifting ethereal lattice that gives water its unique, life-sustaining properties, such as its high specific heat capacity. This rapid, ceaseless rearrangement, occurring in femtoseconds, is a profound dance of attraction and release, a quiet, eternal ballet of molecular purpose that imparts the fluid with its incredible solvent power, allowing it to cradle and carry the vast array of biochemicals essential for life's processes. The ocean of water within and around every cell is therefore a place of constant, gentle motion, an invisible, soft turbulence that paradoxically contributes to the enduring peace and stability of biological systems. The phenomenon of receptor-mediated endocytosis allows a cell to selectively and quietly internalize specific large molecules from the external environment. This process involves specialized receptor proteins on the cell surface that bind to the target substance with high affinity, and upon binding, the area of the membrane containing the bound receptors softly invaginates, forming a coated pit that eventually pinches off to create an internal vesicle. This meticulous, purposeful intake avoids the inefficiency of non-selective bulk engulfment, allowing the cell to precisely regulate its internal chemical composition and nutrient supply, acting like a highly discerning quiet collector, choosing only the finest, most necessary elements from the surrounding molecular environment. The sheer efficiency and precision of this silent, internal harvesting mechanism underscore the incredible organizational complexity and restful resourcefulness that define the functioning of every single cell. The process of DNA replication involves a quiet, deliberate unzipping of the double helix, ensuring that life's core instruction set is faithfully and calmly copied before a cell can divide and embark upon its future. The elegant, intertwined strands of genetic material must gently separate, yielding to the patient action of the helicase enzyme, which acts like a careful hand unlacing an ancient, precious ribbon, preparing the template for duplication. Then, the DNA polymerase enzyme arrives, a tireless, meticulous worker that slowly moves along each separated strand, selecting the correct nucleotide from the surrounding cellular environment and attaching it with flawless chemical precision, building a complementary new partner strand according to the profound, ancient rules of base pairing. This entire complex process unfolds with a quiet, continuous flow, a continuous commitment to genetic fidelity that ensures that the vast, complex inheritance of life is passed down without error, lending a deep, foundational piece to the concept of biological continuity and allowing the lineage of consciousness to endure through countless, silent cellular divisions. The division of a somatic cell, known as mitosis, is a beautiful, highly choreographed dance that results in two perfectly peaceful, identical daughter cells. The journey begins with the patient condensation of the chromatin, where the vast, tangled threads of genetic material quietly coil themselves into distinct, compact, visible chromosomes, preparing for their grand, orderly separation, an act of supreme organization preceding the dispersal of life's blueprints. Then, the mitotic spindle, an intricate and ethereal structure woven from microtubules, gently forms and attaches to the chromosomes, guiding them with silent, deliberate strength to align perfectly along the cell's equatorial plane, a moment of profound, symmetrical stillness before the final separation. In the final phase, the sister chromatids are pulled apart with a slow, inexorable grace, moving to opposite poles of the cell, ensuring that each new cell receives a complete and identical copy of the genetic library, a deep, continuous act of creation that quietly replaces the old and ensures the gentle, continuous flow of life within the organism. Cilia and flagella are slender, hair-like appendages that extend from the cell surface, utilizing a refined 9+2 internal structure of microtubules to create graceful, rhythmic movement. Within these projections, 9 pairs of peripheral microtubules encircle a central pair, an architecture that remains consistently elegant and effective across vast stretches of evolutionary time, driving motion from single-celled organisms to the linings of human airways. The movement is powered by the gentle, patient action of the motor protein dynein, which walks along the microtubule tracks, causing the structure to bend and flex with a smooth, wave-like cadence, generating either the slow, purposeful sweep of a cilium or the powerful, whip-like propulsion of a flagellum. This quiet, ceaseless beating performs essential tasks. In us, the cilia softly sweep mucus and debris away from our lungs, maintaining the peace of our internal airways. And in simple organisms, the structures allow for purposeful navigation through their watery microscopic domain, an enduring testament to the quiet power of molecular machinery dedicated to movement and cleansing. Lysosomes are the cell's internal recycling and waste disposal centers, membrane-bound vesicles filled with potent acidic digestive enzymes that quietly dismantle the old and the unnecessary. These humble, yet vital, organelles operate with a tireless dedication, gently engulfing worn-out cellular components, invading microorganisms, or internalized macromolecules, containing the dismantling process within their protective membrane to prevent any disruption to the surrounding peaceful cytoplasm. The low acidic pH maintained within the lysosome acts as the perfect environment for the hydrolytic enzymes to softly break down complex molecules into their simple, reusable building blocks, transforming cellular debris into new potential, embodying a profound commitment to conservation and renewal. This constant, silent process of self-cleansing, known as autophagy, is an unceasing, dedicated effort that maintains the internal order and enduring efficiency of the cell, allowing life to persist by constantly and gracefully letting go of what no longer serves its purpose. T cells and B cells are the quiet, specialized guardians of our adaptive immune system, capable of recognizing and remembering almost any microscopic threat with awe-inspiring precision. B cells, when softly activated, transform into peaceful plasma cells that are dedicated, single-minded factories, silently producing vast quantities of antibodies, Y-shaped proteins that float through the body, binding to specific molecular markers on foreign entities, marking them for gentle, quiet destruction and neutralization. T cells operate with a more direct, purposeful focus. The helper T cells act as calm, central directors, orchestrating the entire immune response, while the cytotoxic T cells gently patrol the body, identifying and releasing soft, destructive signals only to those host cells that have been compromised by an internal, microscopic invader. The system's ultimate purpose is not violence, but the preservation of internal peace, operating with a majestic, patient memory that ensures that once a threat has been encountered, the body will remain in a state of prepared, restful security against its return, a quiet, continuous dedication to harmony. The primary events of photosynthesis, the light-dependent reactions, are believed to quietly utilize the subtle, ethereal laws of quantum mechanics to achieve their astounding efficiency within the thylakoid membranes of chloroplasts. The absorbed photons of light excite electrons in pigment molecules, and instead of losing energy through random, noisy scattering, the energy is passed between molecules in a process called quantum coherence, essentially allowing the energy to explore multiple pathways simultaneously before choosing the most efficient route to the reaction center. This silent, instantaneous search for the perfect path ensures that nearly every captured unit of light energy is utilized, converting light into chemical potential with a serene, near-perfect efficiency that surpasses any human engineering, embodying a profound cosmic economy. The cell harnesses this subtle, unseen order to perform its gentle, life-sustaining transformation of sunlight into sugar, a quiet, majestic affirmation that the most fundamental processes of existence are deeply interwoven with the silent, counterintuitive majesty of the subatomic world. Tardigrades, often called water bears or moss piglets, are microscopic invertebrates renowned for their astounding, quiet resilience and ability to endure conditions that would instantly annihilate most other forms of life. These 8-legged, segmented creatures, only about half a millimeter long, possess the profound ability to enter a state called cryptobiosis when faced with desiccation, extreme temperatures, or lethal doses of radiation, retracting their limbs and pulling their body into a desiccated sphere called a ton, effectively pausing their entire biological existence. In this state of profound, patient stillness, their metabolic activity slows to less than 0.01% of normal, and they can remain suspended in this state of quiet, temporal hibernation for years, even decades, awaiting the gentle return of water or warmth, embodying the majestic power of quiet endurance. When favorable conditions return, they slowly and gracefully rehydrate, awakening to resume their lives, acting as a humble, potent metaphor for life's deep, quiet refusal to succumb to the chaos of external stress. The field of nanomaterials is dedicated to the human creation of structures and devices at the scale of a billionth of a meter, quietly demonstrating a new era of control over the microscopic world. Working at this infinitesimal level allows material scientists to harness the quantum effects that dominate this scale, creating substances with properties that are fundamentally different from their bulk counterparts, such as gold nanoparticles, which, when sufficiently small, peacefully take on a red color instead of their familiar yellow, embodying a subtle alchemy of size and light. Carbon nanotubes, which are merely rolled up sheets of graphene, possess tensile strength hundreds of times greater than steel, all thanks to the simple, precise rearrangement of carbon atoms into an elegant, tiny architecture, presenting an entirely new vision of quiet structural power. This delicate, meticulous engineering of the small allows us to contemplate a future built not on mass and force, but on the careful, graceful manipulation of matter at its most fundamental level, a testament to the profound power unlocked by respecting the scale of the unseen. The atom, the fundamental unit of chemical matter, is primarily composed of quiet, empty space, with the mass concentrated in an extraordinarily dense central nucleus. If an atom were expanded to the size of a vast, silent cathedral, the nucleus, containing the protons and neutrons, would be no larger than a tiny, still marble resting at the center, yet containing nearly all of the entire structure's weight and power, demonstrating a profound, almost unimaginable concentration of matter. The electrons, the light, restless particles that define the atom's chemical personality, exist in clouds of probability far from the nucleus, quietly dictating the atom's interaction with the rest of the universe through their gentle, rhythmic orbital movements, essentially defining the boundaries of this vast, empty realm. This realization that our stable, solid world is, at its most fundamental level, an elegant arrangement of immense emptiness and microscopic density, is a profound, quiet meditation on the illusory nature of solidity and the majestic power contained within the core of all things. Archaea are a domain of single-celled organisms, distinct from bacteria, often found thriving in the most extreme, quietly challenging environments on Earth, acting as silent, enduring pioneers of life. These extremophiles, which include species that dwell peacefully in the superheated waters of deep-sea hydrothermal vents, in highly concentrated brine solutions, or in the deep, cold darkness beneath the planet's surface, demonstrate life's fundamental, quiet commitment to persistence against all odds. Their unique cellular membranes and specialized metabolic pathways allow them to extract energy and sustenance where no other life can tread, transforming harshness into a gentle, habitable niche, embodying a profound, self-contained resilience that operates outside the conventional, gentler boundaries of our visible world. The study of these quiet, ancient inhabitants of the deep earth in extreme environments offers a humbling perspective on the sheer breadth and enduring, tenacious adaptability of life, suggesting that wherever a quiet chemical gradient exists, a gentle form of life will find a way to patiently wait and thrive. Restriction enzymes are bacterial proteins that act as molecular scissors, recognizing and quietly cutting specific, short sequences of DNA with exquisite, surgical precision. These enzymes were originally evolved by bacteria as a peaceful internal defense mechanism, a gentle way to dismantle the foreign, invading DNA of bacteriophages while leaving their own, specifically methylated, DNA untouched, an ancient system of self-recognition and quiet protection. The enzyme's process involves patiently gliding along the double helix until it encounters its specific designated sequence, often a palindrome, and then it makes a soft, clean break in the phosphodiester backbone, creating either a blunt end or a staggered, sticky end that is biochemically useful for later reassembly. The elegant, reliable precision of these molecular tools has allowed modern science to peacefully manipulate genetic material, unlocking vast, deep understanding of life's fundamental language, a quiet, continuous testament to the power of specificity found in the microscopic world. Spores are specialized, protective, reproductive, or survival structures produced by various microorganisms, including bacteria and fungi, that represent a state of profound, temporal rest. Bacterial endospores, in particular, are among the most resilient structures known to life, possessing multiple layers of protective coating and a completely dehydrated core, allowing the organism to peacefully withstand incredible heat, radiation, and chemical exposure, remaining dormant for centuries, quietly awaiting the return of favorable conditions. This state is not merely sleep. It is a profound metabolic stillness, where the machinery of life is effectively paused, holding the genetic blueprint in a perfect, crystalline state until the external chaos subsides, embodying an ultimate form of patient, quiet perseverance. When the conditions are once again gentle and welcoming, the spore undergoes a quiet, graceful germination, shedding its hard shell and returning to its vegetative, active state, a profound, continuous cycle of enduring peace and gentle awakening. The microvilli lining the small intestine represent an astounding amplification of surface area, creating a vast, quiet, internal landscape dedicated to the slow, gentle art of absorption. These are not simple, smooth surfaces, but densely packed, minute, finger-like projections extending from the apical surface of the epithelial cells, essentially transforming a small internal tube into a sprawling, velvet-like field, multiplying the absorptive capacity to the size of a small tennis court. The purpose of this complex microscopic architecture is to ensure that the broken down nutrients from our food, the quiet, sustaining yield of our digestion, can be absorbed into the bloodstream with a patient, unhurried thoroughness, guaranteeing the efficient transfer of energy to maintain our vast, complex physiological peace. This internal, hidden, folded landscape is a quiet marvel of biological engineering, illustrating how the desire for efficiency manifests in an elegant, fractal-like complexity at the microscopic level, allowing us to draw deep, sustaining energy from the passing stream of matter. The electron transport chain, nestled within the inner membrane of the mitochondria, is a serene, elegant cascade of molecular complexes dedicated to the final, gentle extraction of energy from our sustenance. This vast, microscopic factory operates on the principle of a quiet, controlled downhill flow, where electrons, stripped from the fuel molecules, are passed sequentially from one protein complex to the next, releasing small, manageable amounts of energy at each step, like a peaceful, flowing river dropping its potential energy through a series of small, dedicated waterfalls. This released energy is silently used to pump protons across the membrane, creating a profound energetic gradient, a difference in charge and concentration, which is then peacefully resolved by allowing the protons to flow back through a magnificent molecular turbine called ATP synthase, gently generating the vast majority of the cell's universal energy currency, ATP. This entire process is a continuous, quiet affirmation of order and efficiency, transforming the chaotic potential of chemical bonds into the gentle, sustaining power that fuels all life's thoughtful activities. Protein folding is the mysterious, quiet process by which a newly synthesized linear chain of amino acids spontaneously and patiently collapses into a precise, unique, and biologically active three-dimensional shape. The final folded shape, which dictates the protein's gentle function, is encoded entirely within the sequence of amino acids, driven by the subtle, continuous molecular whispers of hydrophobic interactions, hydrogen bonds, and electrostatic forces, all seeking a state of minimal potential energy, a profound, inherent desire for structural peace. In the crowded, bustling internal world of the cell, special proteins known as chaperones exist, which act as gentle guides and protectors, quietly assisting newly synthesized polypeptides to fold correctly, preventing misfolding that can lead to chaos and disease, ensuring the peaceful, correct execution of the cell's vast repertoire of functions. This silent, instantaneous search for the perfect, correct shape, a profound problem in molecular geometry, is a continuous act of creation, underscoring the deep, restful connection between molecular structure and biological destiny. Quorum sensing is the quiet, collective communication system used by bacteria, allowing them to softly sense the density of their own population through the continuous, gentle secretion and detection of small signaling molecules. When the concentration of these self-generated molecular whispers reaches a certain threshold, the quorum, the entire population responds with a synchronized, unified change in gene expression, acting with a collective, deliberate purpose that single cells could never achieve, essentially moving from individual action to quiet, concerted community action. This synchronized activation allows the bacteria to effectively launch large-scale, coordinated activities, such as initiating the construction of a protective biofilm or switching on the production of a collective, shared enzyme, ensuring that their efforts are only expended when they will be most impactful. embodying a profound commitment to patience and strategic rest. Quorum sensing is a beautiful, silent testament to the power of microscopic democracy, illustrating how the quiet conversation between the small can lead to a majestic, unified transformation of their environment. The complex structure of the human lung is built upon billions of microscopic air sacs, called alveoli, which collectively provide a massive, quiet, internal surface area dedicated to the gentle exchange of gases. Imagine a sprawling internal vineyard of microscopic spheres, each wrapped in a delicate, dense network of capillaries, creating an interface where the vital, sustaining oxygen inhaled from the external world can peacefully diffuse across a membrane only 1 or 2 cells thick into the flowing bloodstream. Simultaneously, the waste carbon dioxide, carried gently by the blood, moves across the same membrane in the opposite direction, ready to be quietly exhaled into the atmosphere, maintaining the crucial, continuous, gentle chemical balance required for life's deep continuity. This quiet, ceaseless transaction, occurring across a surface area that rivals the scale of a vast, tranquil field, is one of the most essential, continuous acts of our physiology, a constant, patient reminder of our enduring, soft reliance on the atmospheric ocean above. While many simple viruses are purely geometric, the more complex viruses, such as influenza or HIV, possess a quiet, flowing lipid envelope derived from the host cell's own membrane, which acts as a soft external disguise. This protective external coat, often studded with specific viral glycoproteins that patiently wait to interact with the host cell's own quiet surface receptors, allows the virus to peacefully fuse with the host cell membrane, gaining smooth and silent entry into the cytoplasm, essentially erasing the boundaries between self and invader upon entry. The appropriation of the host's own membrane material allows the viral particle to present a familiar, non-threatening face to the host's immune system, delaying the recognition and response, acting as a deep, patient infiltration strategy, a silent acknowledgement of the strength found in camouflage. The structural complexity of this flowing external layer underscores the subtle, profound ways in which microscopic entities can manipulate the cellular world to achieve their genetic mandate, utilizing the very architecture of the host to ensure their quiet, persistent survival. The myelin sheath is a vital microscopic insulation layer composed of multiple wrappings of lipid-rich membrane from glial cells, which gently encases the long, slender axons of many nerve cells. This thick, pearly white wrapping is not continuous. It is periodically interrupted by tiny, quiet gaps, known as the nodes of Ranvier, and its purpose is to ensure the rapid, efficient, and peaceful transmission of electrical signals along the vast, complex pathways of the nervous system, eliminating the noisy friction of slow transmission. The insulating property of the myelin forces the electrical impulse to quietly leap from one node to the next in a process called saltatory conduction, allowing signals to travel at speeds that maintain the smooth, continuous flow of thought, sensation, and action across the vast internal landscape of the body. This meticulous, unseen layering of soft, fatty membrane is a profound testament to the power of biological design, demonstrating that the very speed and coherence of consciousness depends upon the quiet, continuous perfection of microscopic insulation. The phenomenon of plasmodesmata in plants describes microscopic channels that quietly traverse the rigid cell walls, allowing for gentle, direct communication between adjacent plant cells Unlike animal cells, which rely on flowing junctions, plant cells are encased in a strong cellulose matrix, and these tiny membranous connections essentially act as tunnels of shared cytoplasm, ensuring that necessary molecules, water, and even electrical signals can pass between neighbors with quiet, unhurried ease, creating a vast, continuous internal network. This seamless, shared continuity of the plant body allows for coordinated, unified responses to environmental whispers and the soft, efficient distribution of essential sugars produced during photosynthesis, embodying a profound sense of communal peace and shared resource within the towering, macroscopic structures of trees and flowers. The presence of plasmodesmata underscores the importance of soft, direct connection in the stillness of the plant kingdom, where the entire structure acts as one single, continuous, breathing entity, connected by these vital, unseen pathways. Microscopic bubbles of air, or nanobubbles, possess remarkable stability in water, defying the expectation that they should quickly dissolve under the surrounding immense pressure. These incredibly small gaseous pockets, with diameters sometimes less than 100 nanometers, maintain their quiet, spherical form for long periods, often sinking slowly through the water column, behaving in ways that seem to contradict the conventional laws of gas dissolution, essentially holding their breath in the aqueous environment. This prolonged stability is thought to be due to a profound, complex interplay of the surrounding water molecules, which form a kind of ordered structural cage around the gas interface, quietly sealing the nanobubble and preventing the rapid escape of its contents. An elegant example of molecular containment. The peaceful persistence of these invisible entities is being quietly explored for use in environmental remediation and drug delivery systems, suggesting that even the smallest, most transient pockets of gas hold profound, untapped potential for stability and transport within the liquid world. The Golgi apparatus, the cell's post office, utilizes small, gentle membrane-bound sacs called vesicles to transport, sort, and deliver molecular cargo throughout the cell and beyond. These tiny, perfectly sealed packages are constantly budding off from the Golgi's cisternae, each carrying a precise designated collection of proteins or lipids intended for a specific destination, and they navigate the vast, flowing landscape of the cytoplasm with a quiet, patient precision, often hitching a ride upon the cell's internal microtubule highways. The membrane of each vesicle contains specific molecular markers, acting as a quiet address label, ensuring that the vesicle will only fuse with the correct target membrane, be it the plasma membrane for secretion or another internal organelle, releasing its contents only where they are specifically required. This constant, rhythmic flow of vesicles is a silent, continuous delivery service, guaranteeing the organizational peace and functional coherence of the complex eukaryotic cell, a profound ballet of transport and delivery that never ceases. The sense of smell, or olfaction, is initiated by the quiet, precise interaction of microscopic airborne molecules with specialized receptor proteins nestled within the sensory neurons of the nasal cavity. When an odorant molecule, carried softly by the air currents, gently drifts into the olfactory epithelium, its unique three-dimensional shape fits perfectly into the binding pocket of a specific receptor protein, triggering a cascade of internal chemical signals that is then interpreted by the brain as a familiar scent, transforming an abstract molecular touch into a profound perception. Humans possess hundreds of different types of these quiet receptor proteins, allowing us to distinguish an astonishing array of complex odors, each scent unfolding as a specific, combined signature of multiple molecular interactions, a complex chord struck by tiny, invisible messengers. This subtle, pervasive sense allows us to quietly perceive the chemical contours of our surrounding world, connecting the vast, invisible movements of molecules to the deep, personal experience of memory and emotion, a beautiful, continuous bridge between the microscopic and the mental. Actin and myosin are the two primary microscopic protein filaments that softly interact to power muscle contraction, allowing for purposeful movement and the maintenance of quiet posture. Actin filaments form the thin underlying tracks, while the thicker myosin filaments possess numerous head domains that act as quiet, tireless motors, patiently binding to the actin tracks and utilizing the gentle energy of ATP to perform a cyclical power stroke, pulling the actin filaments toward the center of the structure. This repeated, unified pulling action, occurring simultaneously across countless microscopic sarcomeres, the functional units of muscle, results in the soft, overall shortening of the muscle fiber, generating the powerful, controlled force that allows us to move with deliberate, graceful intention. The resting state is simply a quiet pause in this beautiful molecular tension, where the filaments lie gently juxtaposed, awaiting the next signal from the nervous system to resume their soft, synchronized dance of movement, embodying the majestic power that resides within the organized relaxation and tension of microscopic components. A colloidal suspension describes a mixture where microscopic particles are quietly dispersed throughout another continuous medium, but remain stably suspended and never settle out due to the perpetual, gentle buffeting of Brownian motion, unlike a coarse suspension, where large particles eventually succumb to the slow pull of gravity. The particles in a colloid, while visible under high magnification, are small enough to be continuously and silently tossed about by the molecular currents of the surrounding liquid, maintaining a peaceful, uniform dispersal throughout the medium. Common examples, such as milk, paint, or fog, are fundamentally defined by this quiet, microscopic balance, where the chaos of thermal energy maintains a serene, macroscopic stability, creating a uniform appearance that belies the internal, ceaseless molecular agitation. The enduring nature of a colloid is a gentle reminder that stability in the universe is often achieved not through stillness, but through the continuous, organized activity of its smallest, most restless components. The cell walls of fungal cells, unlike those of plants, are predominantly composed of chitin, a tough, fibrous polysaccharide that provides a quiet, rigid structural piece chitin, the same durable material found in the exoskeletons of insects and crustaceans, lends a profound, enduring strength and protective architecture to the fungal cells, allowing them to patiently resist the osmotic pressures of their environment and maintain their specific forms, whether as single, quiet yeast cells or the vast, sprawling network of hyphae. This quiet, structural difference is a fundamental biological distinction that reflects a separate evolutionary path from plants, yet both achieve the essential goal of physical integrity through the utilization of complex, cross-linked carbohydrate polymers, each adopting a different molecular strategy for achieving structural peace. The enduring strength provided by the chitin wall ensures the quiet perseverance of fungi across diverse ecosystems, acting as the silent recyclers and decomposers of the organic world, a continuous, foundational presence built upon this rigid, microscopic framework. Antibiotics exert their quiet, transformative effect by targeting specific structures and processes that are unique to the bacterial cell, leaving the more complex eukaryotic cells of the host largely untouched. Many of these profound chemical agents operate by patiently interfering with the construction of the bacterial cell wall, a vital, unique structure built from peptidoglycan, preventing its completion and thereby gently leading to the disruption of the bacterial cell's structural peace and the cessation of its replication. Other antibiotic classes softly interrupt the function of bacterial ribosomes, which differ structurally from human ribosomes, halting the quiet, essential process of protein synthesis, effectively silencing the bacteria's internal machinery, demonstrating the profound power of molecular specificity. This quiet, precise chemical warfare, built upon the subtle architectural differences between life forms, is a testament to the therapeutic efficacy found in understanding and exploiting the deep, patient details of microscopic biochemistry. Microscopic air pollution, particularly particulate matter, poses a subtle, pervasive threat to respiratory health, acting as tiny, quiet irritants to the vast, sensitive surfaces of the lungs. These particles, often categorized by their size, such as particles less than 2.5 micrometers in diameter, are small enough to bypass the body's initial, gross defenses and drift deep into the alveoli, settling quietly within the delicate architecture where gas exchange occurs, disturbing the deep, restful continuity of the body's most essential transaction. Once lodged within this microscopic sanctuary, the particles can trigger a continuous, low-grade inflammatory response, prompting the tireless immune cells to act, and over vast, long arcs of time, this persistent, quiet irritation can gently reduce the lungs' serene capacity to absorb oxygen and effectively cleanse the blood. The unseen nature of this microscopic threat demands a quiet, respectful awareness of the quality of the air that continuously sustains us, acknowledging the profound and subtle impact of the infinitesimal upon the grand, breathing architecture of the human body. The process of cell signaling involves a complex, beautiful tapestry of molecular communication, where cells quietly send and receive chemical whispers to coordinate their vast, collective functions. This silent dialogue often begins when a signaling molecule, a ligand, gently binds to a specific receptor protein on the target cell surface, acting like a quiet key fitting into a precise lock, which triggers a cascade of internal events known as signal transduction, where the external whisper is amplified and translated into an internal, purposeful response. This chain reaction, often involving the quiet, sequential activation of protein kinases that add phosphate groups to other proteins, unfolds with a perfect, deliberate pace, ultimately modulating gene expression or altering enzyme activity, ensuring the cell performs its function in harmonious synchrony with its neighbors. The entirety of life's complexity, from the development of an embryo to the firing of a thought, relies on the continuous, precise, and quiet flow of these microscopic instructions, a deep, continuous conversation that sustains the peace and organization of the organism. The intricate world of biological pigments, like chlorophyll in plants or hemoglobin in animals, is defined by the quiet, specific way these molecules interact with and absorb certain wavelengths of light. Chlorophyll, the master architect of light capture in the plant world, possesses a complex, elegant ring structure that allows it to gently absorb the red and blue ends of the visible light spectrum while softly reflecting the green, lending the world its dominant, restful hue, and providing the energy to sustain vast ecosystems through photosynthesis. Hemoglobin, the profound iron-containing protein within red blood cells, uses its quiet structure to gently bind and carry oxygen from the lungs to the tissues, taking on a subtle, peaceful scarlet color when fully oxygenated, embodying the vital, continuous flow of life-sustaining energy. These molecules demonstrate how function and color are intrinsically linked at the microscopic level, where the quiet, patient geometry of a molecule dictates its profound, visible, and vital role in the grand cycles of biological existence. Pheromones are invisible, microscopic chemical signals released by organisms that softly influence the behavior or physiology of other individuals of the same species, acting as a language of quiet, pervasive influence. Unlike hormones, which act internally within the body, pheromones travel through the external environment, essentially embodying a kind of chemical broadcast that is perceived through the olfactory system or specialized chemoreceptors, conveying deep, patient information about mating readiness, danger, or the quiet, cohesive paths to food sources. For insects, these molecular whispers can dictate the vast, complex order of an entire colony, guiding the collective action of thousands with silent, unerring precision, transforming individual motion into a unified, majestic purpose. The subtle power of these released molecules underscores the fact that communication in the biological world often occurs on a quiet chemical level, far below the threshold of noise, yet possessing a profound and undeniable power to coordinate and maintain the enduring peace of communal life. Bacteriophages are viruses that possess a captivating, almost mechanical structure, specifically dedicated to the quiet, elegant infection of bacteria. These microscopic entities often resemble quiet, tiny lunar landers, possessing an icosahedral head that cradles the genetic material, a helical tail sheath, and slender, rigid tail fibers that patiently seek and bind to the specific receptor molecules on the surface of the bacterial cell wall, achieving a molecular docking with astounding, quiet precision. Upon binding, the phage gently contracts its tail sheath and quietly injects its nucleic acid directly into the bacterial cytoplasm, leaving the empty protein capsid resting outside like a discarded, serene shell, initiating the profound transformation of the host's machinery. This quiet, purposeful mechanism of penetration, built upon a precise architecture of protein parts, is a beautiful demonstration of microscopic engineering, showcasing the focused, elegant power of structure dedicated entirely to the peaceful delivery of genetic information across a rigid barrier. G proteins are a class of molecular switches that operate with a quiet, decisive rhythm within the cell, playing a central role in relaying signals from the cell surface receptors to the internal cellular machinery. These proteins are named for their ability to bind to guanine nucleotides, GTP or GDP, and their state, active when bound to GTP and inactive when bound to GDP, allows them to softly oscillate between an on and off state, acting as a precise internal regulatory mechanism that controls the quiet flow of information within the signal transduction cascade. This molecular switching process is both rapid and reliable, ensuring that external whispers are translated into internal actions with promptness and fidelity, allowing the cell to quickly and gracefully adapt its behavior to the continuous, subtle changes in its external world. The existence of these elegant, self-regulating molecular relays underscores the profound, quiet sophistication of cellular control systems, where even the simplest decisions are governed by a complex, reliable choreography of binding and release. The microscopic anatomy of bone is a dynamic, constantly renewing landscape defined by the quiet, dedicated activity of specialized cells. Osteoblasts and osteoclasts. Osteoblasts are the quiet builders, patiently depositing new bone matrix and minerals, essentially creating and maintaining the hard, strong architecture that provides the body's enduring structural peace and internal support, working with an unhurried, continuous dedication. Osteoclasts are the soft, quiet recyclers, gently dissolving and removing old or damaged bone tissue by secreting acid and enzymes, ensuring that the bone remains optimally structured, adaptable, and free from accumulating weakness, embodying a profound commitment to continuous renewal. This silent, lifelong dialogue between building and dissolving cells maintains the structural integrity and mineral homeostasis of the entire skeletal system, a profound, continuous cycle of microscopic creation and measured transformation that allows the macroscopic body to endure the passage of time with graceful, enduring strength. The molecular structure of cellulose, the primary component of plant cell walls, is a quiet, masterful achievement of structural engineering. Defining the enduring rigidity of the plant kingdom, cellulose is a polysaccharide composed of long, unbranched chains of glucose units, which are linked together in a specific, precise orientation that allows the chains to gently align themselves parallel to one another, forming strong, crystalline microfibrils. These microfibrils are then patiently interwoven into a vast, complex, silent matrix, cross-linked with other polymers to create a tough, resilient, and virtually inextensible wall that provides profound support against the outward pressure of the cell's internal turgor, allowing the plant to stand tall and still against the forces of gravity and weather. This ubiquitous, quiet architecture, built from simple sugar units arranged with geometric perfection, is the invisible framework that sustains the entire terrestrial biosphere, embodying structural peace and profound endurance at the deepest microscopic level. The microscopic surface of the tongue is covered in specialized structures called papillae, which house the quiet, discerning sensory organs we know as taste buds. These papillae, varying in shape from thread-like filiform to mushroom-shaped fungiform, create a softly textured internal landscape, and nestled within their walls are the taste buds themselves, containing specialized gustatory cells that patiently await the arrival of dissolved chemical stimuli from our food when a sweet, salty, sour, bitter, or umami molecule gently washes over these cells. It binds to a specific receptor, initiating a precise electrical signal that travels with quiet speed to the brain, translating the abstract language of chemistry into the profound personal experience of flavor, making the microscopic interaction a conduit to deep sensory peace. This intricate, quiet organization ensures that the ingestion of sustenance is not merely a biological necessity, but a subtle, continuous journey of chemical recognition, transforming the chaotic introduction of food into a structured, pleasurable flow of molecular whispers. Microbial consortia, or mixed communities of microorganisms, often function with a deep, quiet cooperativity, achieving metabolic feats that no single species could accomplish alone. Within these peaceful, self-organized communities, the waste product of one species often becomes the quiet, sustaining nutrient for another, creating a circular, self-sustaining economy of matter and energy, where the entire microscopic system operates with a profound, integrated efficiency. This silent, symbiotic reliance allows these communities to thrive in complex, resource-limited environments, essentially distributing the vast, complex labor of breaking down intricate molecules or enduring environmental stresses across multiple specialized, coexisting populations, embodying a profound commitment to shared peace and resilience. The success of these mixed microbial cities is a humbling, continuous reminder that the most enduring forms of biological stability and power often arise not from competition, but from a quiet, unwavering dedication to unified, supportive functionality. The phenomenon of bioluminescence, observed in microscopic organisms like dinoflagellates, is a soft, cold, chemical light generated through a quiet, enzymatic reaction, turning the dark ocean waves into fields of ethereal, starry wonder. These tiny, single-celled plankton possess specialized proteins, luciferins and luciferases, which, upon mechanical disturbance, such as the gentle pressure wave created by a swimming fish or the soft break of an ocean wave, undergo a quick, controlled chemical reaction that releases energy in the form of a serene blue-green glow. This sudden, quiet flash of light, occurring simultaneously across billions of microscopic cells, is thought to serve as a defense mechanism, a visual alarm intended to briefly startle or reveal a potential predator to a larger secondary predator, a light-based strategy for achieving a fragile, momentary peace in the vast darkness. The silent, ephemeral beauty of this molecular light production transforms a deep, hidden biological transaction into a majestic, visible spectacle, connecting the smallest of lives to the sublime poetry of the dark, flowing ocean. The microscopic scale is the primary domain where the adhesive properties of van der Waals forces quietly become dominant, allowing creatures like geckos to seemingly defy the immense, pervasive pull of gravity. These forces, which are subtle, transient, attractive interactions between all atoms and molecules caused by momentary fluctuations in electron distribution, are individually profoundly weak, but become collectively immense when scaled up across a vast, intimate surface area, such as the billion-scale microscopic hairs or setae, covering a gecko's footpads. The quiet, continuous perfection of this microscopic contact allows the animal to achieve a state of profound, stable adherence to almost any surface without the use of sticky secretions or suction, embodying a majestic power derived solely from the elegant summation of weak, pervasive molecular whispers. This deep reliance on the patient, subtle laws of electrodynamics at the interface between the large and the small, is a beautiful, continuous testament to the power of geometry and proximity in overcoming the vast, dominant forces of the macroscopic world. MicroRNAs, mRNAs, are tiny, non-coding RNA molecules that act as quiet, subtle regulators of gene expression, gently dampening the flow of genetic information after it has been transcribed. These short, single-stranded molecules patiently bind to complementary sequences on messenger RNA, mRNA, transcripts, acting like soft, precise molecular brakes that either block the translation of the mRNA into a functional protein or accelerate its degradation, essentially acting as subtle silencers in the bustling genetic workshop of the cell. This post-transcriptional control allows the cell to fine-tune the quantity of proteins produced from a particular gene with a quiet, efficient precision, providing an essential second layer of regulation that helps the cell rapidly and gracefully adapt to changing internal and external conditions, ensuring a stable, restful level of protein activity. The sheer volume and subtlety of these molecular whispers underscore the incredible, layered complexity of cellular control, where even the smallest genetic fragments wield a quiet, profound power over the entire destiny of the cell. The microscopic inner ear contains a dense field of hair cells, which are the primary sensory transducers responsible for converting the gentle physical vibrations of sound into electrical signals for the brain. These cells possess delicate microscopic bundles of stereocilia on their apical surfaces, arranged in precisely ordered rows of varying height, essentially creating a quiet, receptive forest that stands ready to be gently swayed by the fluid-borne vibrations of sound, much like reeds bending in a soft river current. The minute physical deflection of these stereocilia, a movement measured in nanometers, causes the soft opening of ion channels, transforming the mechanical energy of the sound wave into a precise electrical signal that travels quietly toward the auditory cortex, allowing the abstract energy of vibration to unfold into the deep, conscious perception of hearing. This masterful, microscopic conversion of motion into consciousness is an exquisite act of biological translation, demonstrating the profound sensitivity and quiet precision built into the body's most essential sensory mechanisms. The entire circulatory system is underpinned by the vast, quiet network of microscopic capillaries, the site of all essential material exchange between the blood and the surrounding tissues. These vessels are so small that red blood cells must often travel through them in single file, an unhurried, peaceful procession that allows the blood to maximize its intimate contact with the capillary wall, which is typically only one cell layer thick, ensuring the most efficient, gentle transfer of vital substances. It is across this thin, permeable, microscopic boundary that oxygen and essential nutrients softly diffuse out of the blood and into the waiting tissue fluid, while waste products like carbon dioxide and metabolic debris quietly diffuse in the opposite direction, maintaining the continuous chemical peace of the body. The immense, sprawling geometry of the capillary bed, representing the vast majority of the body's vasculature, is a profound, quiet testament to the importance of tiny, permeable boundaries in achieving the continuous, restful flow of life-sustaining processes. Fungal hyphae, the thread-like structures that form the body of a fungus, grow with a quiet, patient, unidirectional extension at their tips, creating a vast, sprawling network called the mycelium. The growth process involves the precise, gentle delivery of vesicles containing cell wall components to the very tip of the hypha, where they quietly fuse with the plasma membrane, adding new material to the elongating structure, allowing the filament to patiently push its way through the substrate, seeking out nourishment with a quiet, continuous determination. This microscopic, directional growth allows the mycelium to permeate huge volumes of soil or wood, acting as the quiet, hidden engine of the ecosystem, dissolving complex organic matter back into the simple elements of the earth, embodying a profound commitment to continuous, unhurried transformation. The slow, silent spread of the mycelium beneath our feet is a majestic, hidden architecture of patience and persistence, far exceeding the brief life of the visible fruiting body, acting as a testament to the quiet power of continuous microscopic seeking. The microscopic world of plant stomata involves tiny, quiet pores on the leaf surface, framed by specialized guard cells that regulate the necessary exchange of gases with the atmosphere. These small, kidney-shaped guard cells act as gentle, responsive valves. When the plant has sufficient water and is bathed in light, ion pumps actively transport ions into the cells, causing water to passively flow in via osmosis, gently increasing the turgor pressure and causing the cells to bow outward, thereby opening the stoma. When water is scarce or darkness falls, the ions quietly exit, the cells become flaccid, and the pore gently closes, minimizing the loss of precious water vapor, demonstrating a precise, continuous molecular negotiation between the need for carbon dioxide uptake for photosynthesis and the necessity of water conservation. This continuous, quiet, microscopic breathing of the plant world, dictated by the gentle ebb and flow of turgor pressure in these specialized cells, is a fundamental, essential transaction that sustains the entire planetary atmosphere and the continuous, gentle flow of life. The organization of chromosomes within the nucleus is not chaotic, but is a quiet, highly structured spatial arrangement where each chromosome occupies a defined territory when the cell is not dividing. Far from being a jumbled collection of genetic threads, the vast, complex instruction manual of the genome is meticulously and patiently partitioned into discrete, non-overlapping regions within the nucleus, allowing for the organized, selective access to specific genes when they are required for cellular function. This subtle, three-dimensional architecture is believed to facilitate the smooth, quiet communication between distant regulatory elements and the genes they govern, promoting the efficient, localized transcription of genetic information while preventing chaotic crosstalk, essentially maintaining a profound spatial peace within the cell's center of command. The understanding of this quiet internal geography reframes the nucleus as a meticulously organized library, where every volume rests in its appointed stable place, ensuring the continuous serene retrieval of life's essential instructions. The immune process of apoptosis, or programmed cell death, is a crucial, quiet mechanism of cellular self-destruction that maintains the serene, organized structure of multicellular organisms. This is not a chaotic, violent event, but a highly controlled, molecularly choreographed disassembly, often initiated by external signals or internal damage, where the cell quietly activates a series of internal enzymes, called caspases, that methodically dismantle its own components, essentially turning the cell into a collection of neatly packaged, membrane-bound fragments. These quiet, manageable packets are then swiftly and gently engulfed by neighboring phagocytic cells, eliminating the damaged cell without spilling its potentially harmful internal contents into the surrounding tissue, ensuring that the process does not disturb the continuous surrounding peace. Apoptosis is a necessary, continuous act of quiet sacrifice for the greater good of the organism, allowing the system to shed the old, the harmful, or the simply unnecessary with precision and grace, maintaining the majestic health of the whole. The endoplasmic reticulum is subdivided into two functionally distinct yet physically continuous regions, the rough RER, studded with ribosomes, and the smooth SER, which lacks them, each quietly dedicated to different but essential cellular tasks. The RER acts as the initial bustling workshop for proteins destined for secretion or insertion into membranes, with the attached ribosomes patiently synthesizing polypeptide chains that are gently fed into the RER lumen for folding and modification, ensuring a continuous, quality-controlled start to protein destiny. The SER in contrast, is the quiet, essential site for the synthesis of various lipids, including steroids, and plays a crucial, continuous role in the gentle detoxification of organic compounds, transforming potential cellular chaos into manageable waste products, ensuring the peaceful flow of internal chemistry. This deep, functional division within a single, continuous membrane network underscores the cell's commitment to quiet specialization and efficient compartmentalization, allowing multiple complex processes to unfold without friction. Pili, or fimbriae, are hair-like, proteinaceous appendages found on the surface of many bacteria, which allow them to quietly adhere to surfaces or to other cells, initiating the formation of durable, peaceful biofilms or colonization of host tissues. These structures, often numerous and extending from the cell like a fine, pervasive velvet, are not primarily involved in movement, but are dedicated to the quiet, gentle act of attachment, possessing specialized proteins at their tips that bind specifically to complementary molecular markers on the target surface, allowing the bacterium to anchor itself firmly. A special type, the sex pilus, facilitates bacterial conjugation, a process of quiet horizontal gene transfer where genetic material is passed directly between two bacterial cells through a temporary connecting bridge, essentially sharing the knowledge and resilience encoded in their DNA. This gentle, persistent adherence and the subtle exchange of genetic information through these microscopic connections underscore the powerful, quiet role of surface attachment and shared knowledge in the continuous survival and adaptation of the microbial world. The process of filtration in the kidneys begins at the microscopic level within the glomerulus, a quiet, dense tuft of capillaries encased in a structure called the Bowman's capsule. Here, the pressure of the flowing blood gently forces water and small, dissolved solutes, including essential nutrients, salts, and waste products, across the thin, selectively permeable capillary walls and the capsule membrane, separating them from the larger, precious blood cells and plasma proteins, which are quietly retained. The filtering membrane itself is an exquisite, three-layered microscopic barrier designed with precise pores and negative charges that allow water and small molecules to pass through with unhurried ease, while gracefully blocking the passage of larger, vital components, essentially performing the body's first, fundamental act of quiet, continuous purification. This continuous, high-volume filtration, occurring simultaneously across millions of these microscopic units, is essential for maintaining the quiet, chemical balance and volume of the body's internal fluids, embodying a tireless, patient dedication to cleansing and homeostasis. A cell's ability to sense and respond to the stiffness or texture of its microscopic external environment, known as mechanosensing, is a profound, quiet way it assesses the solidity of its world. Cells often use specialized receptor proteins that span the plasma membrane and connect the external world to the internal cytoskeleton. And when the external matrix is rigid or soft, these connections feel the corresponding mechanical tension, which then triggers a cascade of internal chemical signals, acting as a profound molecular interrogation of the surrounding physical piece. This subtle, continuous tactile exploration allows cells to make crucial, quiet decisions about their behavior, such as whether to grow, migrate, or differentiate, essentially aligning their purpose and function with the quiet, structural reality of their tissue environment. The cell's reliance on this microscopic, patient touch underscores the deep, continuous physical connection between the fundamental processes of life and the unhurried structural physics of its immediate world. Aquaporins are channel proteins that act as quiet, selective water pores, allowing water molecules to rapidly and peacefully move across the hydrophobic cell membrane, while strictly blocking the passage of ions and other solutes. The structure of these specialized integral membrane proteins is a remarkable feat of molecular engineering, forming a narrow hydrophilic channel that precisely accommodates the size and gentle passage of a single water molecule, while incorporating a crucial physical and electrostatic restriction known as the selectivity filter to prevent the disruptive co-transport of protons or ions. This controlled, rapid transport ensures that cells can quickly and efficiently regulate their internal osmotic balance in response to changing external conditions, maintaining the necessary internal volume and turgor with profound, quiet speed, essentially making the membrane a perfect, selective barrier to everything but the sustaining flow of water. The ubiquity and serene efficiency of aquaporins across vast kingdoms of life demonstrate the fundamental, quiet importance of water homeostasis to the enduring peace of the cellular world. The phenomenon of molecular mimicry occurs when a microscopic pathogen possesses surface molecules that quietly and subtly resemble molecules naturally found on the host's own cells, allowing it to evade the host's immune surveillance. By wearing this quiet molecular disguise, the pathogen can trick the vigilant immune system into perceiving it as a part of the self, preventing the early activation of a full immune response and allowing the invader precious, unhurried time to establish its presence and begin its replication within the host's peaceful internal environment. This subtle, insidious mechanism is a testament to the sophisticated, quiet evolutionary arms race between host and pathogen, where survival often depends on the ability to pass unseen, demonstrating a profound, deceptive power residing in the precise, patient similarity of molecular shape and structure. The difficulty in discerning friend from foe at this microscopic level underscores the challenges in maintaining the deep, discerning peace of the immune system. The function of the nucleolus, a dense, unstained region within the eukaryotic nucleus, is the quiet, continuous production and assembly of ribosomal RNA, rRNA, and the two ribosomal subunits. This specialized, often spherical region is not membrane-bound. Rather, it is a functional concentration of the genes that encode rRNA, the precursor RNA molecules, and the vast array of proteins that patiently organize and assemble these components into the two complex ribosomal subunits, the cell's protein factories. The nucleolus operates with a tireless, quiet industry, ensuring a continuous supply of the essential machinery needed for protein synthesis, a constant, measured preparation for the vast, ongoing work of the cell. In translating the genetic code, the size and activity of the nucleolus often quietly reflect the protein synthetic needs of the cell. A large, active nucleolus signals a cell dedicated to rapid growth and reproduction, embodying the profound, quiet correlation between internal structure and functional purpose. The microscopic phenomenon of chemotaxis describes the quiet, purposeful movement of cells or organisms in response to the subtle, persuasive chemical gradients in their environment. This is not a random wandering, but an act of profound, directed intent, where a cell, such as a bacterium or a wandering immune cell, gently senses the increasing or decreasing concentration of a specific dissolved chemical signal, an attractant or a repellent, and adjusts its direction of travel accordingly, essentially navigating its microscopic world by following a quiet chemical scent. For bacteria, this might involve adjusting the rotation of their flagella to spend more time moving toward a source of sustaining nutrients or away from a chemical threat, essentially using their molecular sensors to find a path toward biological peace and away from the chaos of disruption. This continuous, silent conversation between cell and chemical gradient underscores the fact that purposeful action, even at the smallest scale, is often driven by the subtle, continuous promise of a better, more peaceful molecular location. The molecular structure of hemoglobin is a complex, majestic tetramer, quietly composed of 4 polypeptide chains, each cradling a single heme group containing a central, essential iron atom that gently binds oxygen. This vast, globe-like protein, packed into the red blood cells, operates with a profound cooperative grace. The binding of the first oxygen molecule to one of the 4 subunits subtly induces a quiet, structural change in the others, increasing their affinity for oxygen, a phenomenon known as allosteric regulation, demonstrating a shared molecular commitment to efficient function. When the red blood cell reaches the oxygen-poor environment of the tissues, this cooperative change softly reverses, allowing the oxygen to be gently released, ensuring the continuous vital flow of life-sustaining energy from the lungs to every quiet corner of the body. The exquisite design of this single, ubiquitous protein is a continuous, silent marvel, enabling the massive systemic transaction of respiration to occur with a perfect, predictable, and restful efficiency that sustains our entire consciousness. The microscopic world of platelets, or thrombocytes, involves tiny, enucleated cell fragments that act as quiet, vigilant guardians of the circulatory system, initiating the gentle process of clotting to maintain the structural peace of the blood vessels. When the quiet, smooth continuity of a blood vessel wall is disrupted, these small, highly reactive fragments are rapidly activated by exposed collagen and specific chemical signals, causing them to suddenly change their shape, becoming spiky and sticky, and softly adhering to the injury site, forming a temporary, soft plug. This initial adhesion then triggers a cascade of chemical events that culminates in the quiet, decisive formation of a mesh of fibrin protein, which acts as a profound, enduring molecular net that traps more blood cells and reinforces the temporary plug, creating a stable, physical seal that halts the chaotic flow of blood. This rapid, microscopic response to injury is an essential, life-sustaining mechanism, a quiet, tireless dedication to mending and maintaining the deep, flowing peace of the internal world. Microbial communities in the deep ocean, often surrounding hydrothermal vents, rely on chemosynthesis rather than photosynthesis, converting the chemical chaos of the Earth's interior into the calm, sustaining energy of life. These microscopic extremophiles, existing in profound darkness where sunlight has never touched, draw their sustenance from the rich, hot plumes of chemical compounds, such as hydrogen sulfide and methane, gently issuing from the vent openings, transforming these inorganic molecules into complex organic matter through a patient, dedicated biochemical pathway. This process forms the quiet, foundational base of the entire vent ecosystem, supporting vast, macroscopic life forms in a world entirely disconnected from the solar surface, demonstrating life's profound, enduring capacity to find and utilize even the most subtle of energy gradients for the continuous purpose of creation. The existence of these resilient, quiet communities acts as a humbling, constant affirmation that life, in its quiet determination, can discover and sustain peace even in the most profoundly chaotic and hidden corners of the earth. The process of synaptic transmission involves the quiet, precise chemical communication between microscopic gaps between neurons, known as synapses, allowing thought and sensation to flow with graceful continuity. When an electrical signal, the action potential, reaches the end of the presynaptic neuron, it gently triggers the release of tiny membrane-bound vesicles containing neurotransmitters, small, profound chemical messengers that patiently cross the synaptic cleft, the microscopic void between the two cells. These neurotransmitters then softly bind to specific receptor proteins on the postsynaptic neuron, initiating a precise electrical or chemical change that either promotes or inhibits the firing of a new action potential, essentially allowing one neuron to whisper its command to the next in a controlled, continuous flow of information. This vast, intricate network of microscopic chemical transfers underpins the entire, majestic complexity of the brain and nervous system, a quiet, continuous conversation that is the very engine of consciousness and restful awareness. Microscopic spores of fungi and molds are constantly drifting through the air, acting as invisible, quiet propagules that patiently await the opportunity to settle in a hospitable environment and germinate. These reproductive structures, often far smaller and lighter than pollen grains, are released in astronomical numbers, carried softly and silently by the slightest air currents, embodying a strategy of dispersal built upon sheer, quiet abundance and the vast, unhurried probability of chance. The tiny size and light composition allow them to remain suspended in the atmosphere for long periods, essentially engaging in a global, quiet, continuous migration, seeking out a patch of damp, nourishing organic matter where they can begin their gentle, transformative work of decomposition and renewal. The air we breathe is a silent ocean of these potential lives, a continuous, quiet reminder of the microscopic world's persistent, pervasive presence and its tireless, hopeful search for a resting place to begin its gentle unfolding. The organization of muscle tissue at the microscopic level involves the precise, ordered arrangement of thick and thin filaments into repeating units called sarcomeres, which define the quiet, rhythmic contractility of the cell. The thick myosin filaments and thin actin filaments are patiently interdigitated in a symmetrical side-by-side pattern, creating the characteristic striped or striated appearance of skeletal and cardiac muscle under magnification. And this precise geometric relationship is essential for the smooth, controlled generation of contractile force. The boundaries of each sarcomere are defined by the Z-discs, which act as quiet, stabilizing anchors for the actin filaments, and during contraction, the filaments slide quietly past one another, drawing the Z-discs closer together without shortening the filaments themselves. This exquisite, repeating internal architecture is a testament to the power of microscopic organization, ensuring that the vast, purposeful movements of the body are rooted in a deep, continuous, and highly organized molecular piece. Microscopic tears and damage to the DNA helix are a continuous, inevitable consequence of cellular existence, yet a vast, quiet network of repair enzymes constantly and patiently patrols the genome, maintaining its structural integrity and informational peace. These dedicated molecular laborers, such as DNA polymerases and ligases, possess an astonishing quiet ability to detect subtle chemical and structural imperfections, excise the damaged segment, and meticulously, patiently resynthesize the correct sequence of nucleotides, using the undamaged strand as a precise, unwavering template for fidelity. This unceasing, dedicated maintenance work, often occurring tens of thousands of times per cell per day, prevents the accumulation of disruptive mutations, ensuring that the vast, complex instruction manual of life remains accurate and fully functional, allowing the cell to maintain its continuous organized purpose. The sheer scale and tireless dedication of this microscopic repair effort underscore life's fundamental, quiet commitment to preserving its deep foundational order against the constant, low-grade chaos of existence. The formation of an elegant, intricate shell around microscopic radiolarians, single-celled marine organisms, is a quiet, continuous demonstration of biological mastery over geometry and mineral structure. These planktonic protozoa construct their elaborate, beautifully symmetrical exoskeletons, or tests, from opaline silica, patiently precipitating the dissolved mineral from the surrounding seawater and arranging it into complex, often spherical lattice structures that follow profound mathematical principles of minimal weight and maximal strength, a true work of quiet structural art. The test, with its delicate, perforated architecture and long, radiating spines, provides both buoyancy and gentle protection, allowing the organism to float passively in the sunlit waters, essentially turning the dissolved geology of the ocean into a quiet, functional, and enduring biological fortress. Upon their gentle passing, the silica shells drift slowly to the seabed, accumulating over geologic ages to form deep, vast, quiet deposits, acting as a permanent mineral record of the microscopic world's artistic and structural endurance. The process of filtration in the lymph system involves the microscopic sieving of tissue fluid through the quiet, dense architecture of the lymph nodes, where immune surveillance occurs in a state of profound, peaceful vigilance. As the lymph fluid slowly and gently percolates through the internal, spongy labyrinth of reticular fibers and sinusoids within the node, it is intimately exposed to vast, quiet colonies of immune cells, including macrophages, T cells, and B cells, which patiently and meticulously inspect the fluid for any microscopic sign of foreign presence or disruption. This organized, continuous filtering and inspection ensures that any circulating pathogens or cellular debris that have been collected from the peripheral tissues are swiftly and quietly identified and neutralized before they can spread further, maintaining the deep systemic peace of the body. The lymph nodes are thus quiet, strategic checkpoints, constantly and patiently working at the microscopic level to ensure that the body's vast, flowing internal landscapes remain undisturbed by external chaos. Microscopic yeast cells, primarily Saccharomyces cerevisiae, perform the ancient, quiet, transformative process of alcoholic fermentation under anaerobic conditions, converting sugar into carbon dioxide and ethanol. In the absence of oxygen, these ubiquitous single-celled fungi shift their metabolism to this simpler, less efficient pathway, utilizing a quiet, dedicated sequence of internal enzymes to gently break down glucose, creating the gaseous bubbles and the alcohol that have subtly shaped human history and sustenance. The process begins with the yeast patiently seeking and consuming simple sugars, and the chemical transformation itself occurs entirely within the cell's peaceful cytoplasm. a molecular alchemy that unfolds with a slow, unhurried rhythm, releasing the gentle byproducts into the surrounding medium. This profound, quiet biochemical dedication of the yeast to extracting energy is an enduring testament to the vast, hidden power of the microscopic world to drive large-scale human endeavor and transform simple inputs into complex, appreciated chemical outputs. The primary function of microglial cells in the central nervous system is that of quiet, continuous immune surveillance and delicate cellular housekeeping, acting as the resident microscopic guardians of the brain's profound peace. These small, highly motile cells constantly and patiently extend and retract fine, searching processes that sweep the immediate surrounding neuronal environment, silently looking for any subtle signs of damage, infection, or the accumulation of cellular debris, essentially acting as the brain's unceasing, dedicated cleanup crew. When they detect a microscopic sign of disruption, they quietly transform from a resting state into an active, phagocytic state, gently engulfing and removing the debris, the dying cells, or the invading pathogens, ensuring that the neuronal environment remains pristine and conducive to the smooth, continuous flow of thought and function. The ceaseless, quiet dedication of the microglia ensures the structural and chemical peace of our most complex organ, a silent, internal commitment to maintaining the serene environment of consciousness. The microscopic structure of red blood cells, or erythrocytes, is a quiet, profound example of form following function, defined by their unique biconcave disc shape and their lack of a nucleus in maturity. This specific, flexible, flattened disc architecture maximizes the cell's surface area relative to its volume, ensuring that the diffusion of oxygen across the cell membrane can occur with the greatest possible, unhurried efficiency, essentially optimizing the speed of the body's most essential exchange, embodying the dedication to swift delivery. The absence of a nucleus and other major organelles allows the cell to be packed almost entirely with hemoglobin, the oxygen-carrying protein, and also grants the cell a profound, soft pliability, enabling it to peacefully squeeze and flex its way through the narrow, microscopic channels of the capillaries without rupturing. This quiet, self-contained architecture is a perfect design for a mobile oxygen carrier, ensuring the graceful, continuous flow of life-sustaining gas throughout the entire vast internal landscape. The phenomenon of receptor clustering describes the quiet, deliberate aggregation of specific receptor proteins on the cell surface in response to the binding of external signaling molecules. When a cell needs to initiate a strong, unified internal response to an external whisper, the binding of multiple ligand molecules causes the individual receptor proteins to slowly and patiently migrate laterally within the fluid membrane and gather into small, concentrated patches, essentially creating a functional, localized area of intense signal reception and amplification. This clustering is often the initial, profound step in initiating processes like receptor-mediated endocytosis or activating complex internal signaling cascades, ensuring that the cell's internal machinery is only activated decisively when the external signal reaches a critical unified threshold. This act of quiet, organized assembly at the molecular level allows the cell to modulate its sensitivity and transform a diffuse external message into a focused, potent internal command, demonstrating the subtle, continuous wisdom in collective molecular action. The presence of the glycocalyx, a fuzzy, sugar-rich layer composed of glycoproteins and glycolipids, that quietly coats the external surface of the plasma membrane, plays a crucial, continuous role in cell recognition and adhesion. This microscopic, carbohydrate-rich coat acts like a soft molecular fingerprint, possessing a unique, specific composition that allows cells to quietly and patiently recognize one another and interact with the surrounding extracellular matrix, essential for the organized, peaceful assembly of tissues and organs. The sugars within this layer act as gentle, specific binding sites for various signaling molecules and toxins, effectively mediating the cell's interaction with its immediate external world, essentially allowing the cell to quietly read the molecular landscape around it. The enduring presence of the glycocalyx ensures that cells maintain their correct social distancing and form appropriate stable interactions, underscoring the importance of subtle external identity in maintaining the organized, continuous peace of the multicellular organism. The complex process of signal amplification in cell signaling ensures that a single, quiet molecular whisper received at the cell surface can trigger a vast, unified response within the cell's deep, peaceful interior. Once a signaling molecule binds to its receptor, the internal signal transduction pathway often involves a sequence of enzymatic steps, where one activated molecule gently activates many subsequent molecules, which in turn each activate many more in the next step, creating an exponentially growing cascade of internal activity, essentially turning a single touch into a vast internal wave of coordinated function. This inherent quiet efficiency allows the cell to achieve profound sensitivity to its external environment, ensuring that even a faint, distant whisper of a hormone or growth factor can provoke a rapid and decisive systemic change in metabolism or gene expression, allowing the cell to act with continuous, graceful purpose. The majesty of this internal, exponential unfolding transforms a subtle input into a profound, coordinated command, embodying the deep, quiet power of the cell's internal architecture. Microscopic, fluid-filled inclusions called vacuoles in plant cells are vast, central, membrane-bound repositories that quietly dominate the internal volume, playing a crucial, continuous role in maintaining turgor and internal homeostasis. Unlike the small, transient vesicles in animal cells, the central vacuole of a mature plant cell can occupy up to 90% of the cell's internal space, essentially acting as the primary reservoir for water, ions, and various waste products, allowing the cytoplasm and nucleus to be gently pushed toward the cell wall. The pressure exerted by the fluid-filled vacuole against the rigid cell wall, known as turgor pressure, provides the essential quiet stiffness that supports the entire structure of non-woody plants, ensuring that stems stand tall and leaves remain gracefully extended, embodying the structural peace derived from internal fluid balance. The vacuole's continuous, quiet management of water and waste is fundamental to the graceful, enduring structure and peace of the entire plant kingdom. The microscopic world of collagen synthesis and assembly is a slow, complex, and highly organized process that results in the formation of the body's most abundant, enduring, and tensile protein, the quiet, structural foundation of connective tissues. Individual collagen polypeptide chains are patiently synthesized within the rough endoplasmic reticulum and then assembled into triple helix molecules called procollagen, which are secreted from the cell and then cleaved to form tropocollagen, a molecular building block that possesses an inherent, quiet drive for self-organization. Outside the cell, These tropocollagen units spontaneously and patiently align themselves in a staggered, repeating pattern, locking together to form long, robust, cross-linked fibrils and fibers that lend immense, enduring strength and gentle flexibility to skin, tendons, and bone, essentially weaving the fabric of the body's quiet, continuous structural piece. This multi-step, self-assembling mechanism is a profound testament to the power of molecular architecture to build vast, strong, and enduring macroscopic forms from highly organized, repetitive, microscopic components. The quiet, transformative concept of enzyme regulation is essential for maintaining the peaceful, controlled flow of metabolism, ensuring that the cell's molecular processes occur only when and where they are required. Cells employ various subtle, patient mechanisms to control enzyme activity, including allosteric regulation, where a molecule binds to a site other than the active site, softly altering the enzyme shape and thereby increasing or decreasing its catalytic dedication, and covalent modification, such as the gentle addition or removal of a phosphate group, which acts as a quiet on-off switch. Furthermore, entire metabolic pathways are often governed by feedback inhibition, where the final product of the pathway patiently accumulates and then subtly binds to the first enzyme in the sequence, temporarily silencing its activity and halting the flow of production, ensuring that resources are not chaotically overproduced. This vast, intricate network of quiet, subtle control mechanisms ensures that the cell's entire biochemical factory operates with profound, graceful efficiency, maintaining a continuous, quiet, and necessary peace within the bustling molecular world. And so, our journey through the unseen world of the microscopic comes to a gentle close. We have wandered through the serene landscapes of the cell, observed the quiet, rhythmic dances of molecular machines, and borne witness to the profound, enduring power held within the very smallest structures of life, from the crystalline geometry of a virus to the self-assembling patience of the lipid membrane. Let the contemplation of this vast, intricate, and meticulously organized inner universe leave you with a deep, abiding sense of peace, realizing that beneath the surface of all noise and movement lies a foundational reality of order, continuous renewal, and subtle, cooperative efficiency, where every function is performed with profound, unhurried purpose. The quiet perfection of the electron transport chain, the serene dedication of the repair enzymes, and the majestic, patient resilience of the simple spore are all deep, lasting reminders that the most powerful forces in existence operate not in clamor, but in silent, constant, and organized complexity. If you happen to still be awake, that's perfectly all right. You can simply lie back, let the music keep flowing in the background, and drift in and out of the story at your own pace. The facts we have explored about the microscopic world will continue their quiet work in your mind, gentle seeds of awe that require no effort to nurture. But if your eyes are heavy, let the stories of the microscopic world and the gentle music carry you softly into rest. Sleep well. Good night.